Hi guys, welcome back to another episode of Champions Guide to Not effing up A-level biology. Today we're going to be doing the light dependent reactions. This is my third take on this video and I'm so fed up right now, but let's hope third time's charm, shall we? So this topic is going to be a pain in the ass for any of those that don't do chemistry, because this topic is very biochemical. And honestly, just suck it up and just learn it, okay? The first two parts, which is the photosynthesis parts, as long as you know them, you're going to be fine for the rest of the topic. It's all just applying the same concept over and over again. Now, I know respiration is a topic that seems like a big step up from GCSE level, and you're not wrong. A lot of the concepts you'll learn in this topic is going to be quite new, and you do have to put a lot of thought into it, but I think it's pretty interesting, photosynthesis and respiration in general, and it really lets you delve into the really molecular parts of biology, which is something that's not really covered too much. Anyway, let's begin. So before we actually go into the light-dependent reactions, I'm going to talk about a few basic principles and things you just need to know in order to understand this topic properly. The first thing are these things called electron carriers. So, electron carriers. Now in textbooks you'll see this as NAD, FAD, maybe even FADH2, NADH, it keeps going over and over again, NAD+. And what electron carriers are, they basically, it's in the name really. They basically carry electrons from one place to another. Now you can't have a bunch of rogue electrons in our body just going all over the place. That'll cause like a lot of chaos. So electron carriers are effectively there to put use to those electrons. You'll find out in this topic that electrons are crucial for generating ATP, which is why electron carriers play such a significant role in what's going on. Fun fact, NAD stands for nicotinamide adenine dinucleotide. So when I first did this topic, I thought NAD was a protein, but that's not actually true. NAD is just a chemical molecule, and it doesn't really behave like a typical protein would, obviously. But yeah, I just found it really interesting how this one chemical is just really important for maintaining just a normal cell, really. So that's what it is. I know there's a lot of variation, like NAD+, and sadly there is no like international convention to writing it. A lot of the stuff just mean the same thing. The way I'll be doing it in my videos is it'll be by almost like one convention, and it doesn't really matter in the end. As long as you stick to one convention, it all should work out. Now, more specifically relating to this video itself, the light-dependent reactions, we're going to talk about something called photosystems. Photosystems. And I'm going to draw two photosystems here. I'm going to call this photosystem 1 and this one photosystem 2. Now, photosystems are, the way I like to think of it is condensed chlorophyll. It's like almost little parcels of chlorophyll and other pigments you can have that allow the plant to absorb different wavelengths of light. So one photosystem will absorb one particular wavelength of light, the other photosystem will absorb a completely different wavelength of light. I also want to make it explicitly clear that photosystems don't just contain chlorophyll. Leaves can be a wide range of colours, and it's because of the different pigments that they have. So some plants might have xanthophylls, which gives you that orangey yellow colour. And again, the reason for this is that plants, so light itself, it comes in a lot of different wavelengths, and by having a wide range of photosystems, it allows the plant to absorb as much light energy as possible, which is crucial for things like photosynthesis, respiration, so for things like, well, not really respiration, more photosynthesis, but nevertheless, all of that light energy is crucial for the plant's growth, and it's why photosystems are very crucial things. The last thing I want to talk about is where photosynthesis actually happens. Now, I see that students really don't get this topic if they don't get the basics right. So I'm going to draw out a chloroplast here. Now within the chloroplast, from topic 2, we have these little discs. Each disc is called a thylakoid, and they have a membrane around them. A stack of thylakoids is called a granum, and the fluid surrounding the thylakoid is called a stroma. Now, you need to get this into your brain. It's like a daily mantra kind of thing, as Mr. Greenberg says. The light-dependent reactions, LDR, happen in the thylakoid membranes, and the light-independent reactions happen in the stroma. Keep saying that in your brain. I can't emphasize this enough. This is really where things get a bit tricky, and if you don't know the exact location of where certain processes are happening, you're not going to be able to do this topic. Light-dependent reaction in the thylakoid membrane, light-independent reaction in the stroma. Alright, so those are the 3 main things, like the principles that you need to know to understand this topic. Next thing we're going to do is the hard part, which is going to be the thing everyone dreads, the light-dependent reaction itself. So the first thing I'm going to do is draw out a phospholipid bilayer. And I'm obviously not going to spend my whole life drawing little phosphate heads because that's annoying. So imagine these are a bunch of phosphate heads and you have those non-polar fatty acid tails, and you form this phospholipid bilayer. I'm going to start labeling the different parts now. This is going to be called the thylakoid space. Thylakoid space. And this is going to be called the stroma space. Now, I'm going to draw a little box here. It's completely separate from this diagram. But I really want you to understand, if this is the thylakoid, this is the thylakoid membrane, and this is the stroma. I really need you to get that into your brain. Alright, the next thing I'm going to draw are those photosystems we were talking about earlier. So I'm going to create a few little spaces here and there. This can be my first photosystem, and this can be my second photosystem. Now I'm going to call this PS2, I'm going to call this PS1, and you'll find out in a bit why that is the case. The actual, well, we're going to go over it, but I think one was discovered earlier than the other, but then biologists have a really weird way of naming stuff, so yeah, I don't think photosystem 1 was the first system that was discovered, but anyway. So I have my photosystems over here. The next thing I'm going to draw are something called H+ pumps. And a pump is basically something that, well, it's kind of self-explanatory. It moves something from one space to another. The crucial part is that this process involves ATP, so it's going to be an active transport kind of process. Or does it involve ATP? Find out in a bit. So I'm going to draw out another H+ pump over here. So I'm going to call this, I'm going to draw it in a, no, it's fine, let's draw in red. So I'm going to call this H+ pump. This is also a H+ pump. And the final thing I'm going to draw out is this weird thing over here. Now, I personally find this one of the most beautiful kind of protein things, and it's going to be familiar. It's called ATP synthase, and we'll see how that works in a bit. So, ATP synthase. So, let's start off from the beginning. Let's start off with our photosystems here. Now, chlorophyll contains electrons, so I'm just going to write that here. Chlorophyll, or actually a better colour might be black, just to distinguish what's going on, because this is going to be a very colourful diagram and there's a lot of stuff that's going to be going on. So I'm going to say 1, chlorophyll contain electrons. Now let's draw out a little sun over here. And plants photosynthesize by using light, right? So that light energy is going to be reaching photosystem 2. Photosystem 2 is the start of the electron transport chain. So all of that light energy reaches photosystem 2. Now, what that light energy does is that it causes electrons in photosystem 2 to be excited. So light energy causes electrons in photosystem 2 to be excited. Now, this energy is so high that it causes an electron to actually fling off from the chlorophyll molecule. And that part is called photoionization. So I'm going to write that down in red. Causes chlorophyll to be photoionized. That's a key term that you kind of need to know. So I'm going to draw out, what colour should I do? Let's do blue. So that energy causes electrons to be lost from the chlorophyll, and we say that the chlorophyll is oxidized. So chlorophyll oxidized. Now this is the chemistry part. Oil rig. Oxidation is lost, reduction is gained. So oxidation, because that chlorophyll molecule is losing electrons, therefore it is oxidized. So that's the first step of what's happening here. Now, these electrons, they're being lost, and we need to replenish those electrons. So what happens is the light energy is also quite powerful to split water. And that process is called photolysis. So I'm going to put 2 here. Let's put it here. So I'm going to write it down in here in blue. So photolysis, light energy, splits water to electrons, H plus, and oxygen. So I'm going to write out the chemical equation here. If you write out the chemical equation, they actually give you a mark for that. So and it saves you a lot of time, so I do that just to be sure. So H2O goes to 2H plus 2 electrons plus a half O2. So that's the chemical equation over there. So all of that light energy is exciting the electrons in the photosystem, and those electrons are at a higher energy level now. And because those electrons are being lost from the chlorophyll, those electrons need to be replenished, and that's carried out by photolysis. The next step we're going to have is that we're going to have a few electron carriers, and I'm going to draw them like this over here and here. Now, you don't need to know the name of these electron carriers in particular, but out of interest, one of them is called plastoquinone, which moves electrons from one place to another, because that's the definition of an electron carrier. So that electron carrier is going to bind to that electron, so it's going to go wee over there, and it's going to move the electron from here through the H plus pump. Now, the energy that electron has, that energy is used to transport hydrogen ions from the stroma space into the thylakoid space. This is where things get really weird. I know people start getting really confused, so I'm going to go really slowly at this stage. So I'm going to have a few H plus here. H plus, H plus, H plus. And the energy from that electron is being used to power the H plus pump, and it causes hydrogen ions to move from the stroma space into the thylakoid space. So I'm going to show that like this. So those protons, hydrogen ions, remember a hydrogen ion is just a proton. I'm going to use those interchangeably because they do that in the exam as well. So H plus over here. So the next thing I'm going to write down is that the energy from excited electron is used to actively transport H plus into the thylakoid space. Remember this diagram over here? From stroma space via your H plus pump. Now, because that energy is being used, the electron is going to actually lose some energy and it's going to go to a lower energy level. So I'm going to symbolize that with a green electron. So it's at a lower energy level because it's used up some energy to pump that H plus pump. Now, the electron carrier over here, I think it's called cytochrome B6. Again, you don't need to know about that, but I just like this topic, so yeah. So the electron here is gonna go wee. It's gonna bind to that electron carrier over there, and it's gonna reach photosystem 1. Now, remember, there's still a lot of light energy, and it's not just going to the photosystems here. It's gonna reach the whole membrane, right? So the light energy from there is also starting to reach photosystem 1. So any electrons here are again going to be excited to a higher energy level. So next thing I'm going to put is that electron carriers move electrons from low energy level to PS1, where light energy re-excites electrons to a higher energy level. So I'm going to symbolize that with a blue electron because we're moving them up back to a higher energy level. So again, the same thing's happening, right? That electron is going to that electron carrier. It's going to use that energy to pump that H plus pump. And again, it's going to actively transport more protons into the thylakoid space. So again, there we go. Into the thylakoid space it goes. Now, after powering that second H plus pump and after photosystem 1, the electron reaches a molecule called, drumroll, NAD+. NADP+, that is, because we're talking about photosynthesis here. Whenever we're talking about photosynthesis and we're talking about electron carriers, always add a P. P for photosynthesis, alright? So that electron is going to reach NADP+. Now, after it gains that electron, it becomes reduced NAD. Remember, oil rig, oxidation is loss, reduction is gain. And electron carriers, they can gain stuff, they can lose electrons. In this case, we're gaining the electron and we're forming NADPH. You might see this being referred to as reduced NAD. So watch out. Now, NADP+, sorry, it can actually hold 2 electrons. So I haven't really seen an exam question where they've ever assessed on that. Maybe I'll find one soon because 4 weeks until exams, everyone's rushing to do past papers. But yeah, just be wary that NADP+ can carry 2 electrons rather than the one that we're illustrating here, but we are simplifying a lot. So I'm going to put 0.5 here. The electron, so I think a better way to rephrase that is that NADP+ is reduced to NADPH. And we're going to specifically say that NADP+ is the final electron acceptor. Final electron acceptor. Because after this, we're going to move on to the light independent reactions. So it is going to be that final electron acceptor. It's going to be the final thing accepting the electron. So Yeah, it's not going to be used for anything else. The final step is that I know all of you guys are thinking, why are we pumping these hydrogen ions? And these hydrogen ions, these hydrogen pumps, they've been working really hard and they've been moving all of this H plus all the way to the thylakoid space. So what's the point of that? Why are we doing that? And that's because we've formed something called a chemi-osmotic gradient. Chemi meaning chemical, osmotic, the movement of... Now I know this is confusing. Osmosis, thinking about water potential and stuff, but osmosis really refers to pressure and changes in concentration. I know it's really weird, but it's called chemiosmosis. And what you can see here is that because of these hydrogen pumps, we now have a really high concentration of H plus in the thylakoid space, but a low concentration of H plus in the stroma space. And these hydrogen ions, they can only move through one channel protein, and that's called ATP synthase. So it's going to be moving from a high concentration to a low concentration. It can't move down the protein system, it can't move down the hydrogen pump. It has to move down via ATP synthase. Now, by moving down ATP synthase, it causes ATP synthase to change its tertiary structure slightly, and that change in tertiary structure causes our friend ADP and inorganic phosphate to turn into ATP. We've been wondering, what's the point of all of these hydrogen ions? And that end goal is to form ATP. To form the ATP that plant needs to carry out all of its metabolic processes. So I've run out of space, so I'm going to try and smoosh it on over here. So we were at 4, 5, 6. Chemiosmotic. This is a key word you really need to write in your exam. Chemiosmotic gradient created between thylakoid space and stroma space. The hydrogen ions will move from a high concentration to a low concentration via ATP synthase. I'm really sorry, I ran out of space. Synthase. That causes a change in tertiary structure, which causes the phosphorylation of ADP into ATP via the inorganic phosphate. Remember what phosphorylation means? Phosphorylation means adding a phosphate group to something, and in this case, it's your friend adenosine diphosphate. And we're forming a bunch of ATP throughout this process. So you might also be thinking, what's the point of producing all of these NADPHs? And we'll find out in the next video when we go over the light independent reactions. Now, the final thing that I want to talk about, I know this video is getting long and I'm really sorry about that, but this is how this topic is. So, yeah. The last thing I'm going to talk about is cyclic and non-cyclic photophosphorylation. This whole process is called the electron transport chain, or the chemiosmotic theory. It might be how it's labeled in the textbook, for example. This process is non-cyclic photophosphorylation because it involves photosystems 2 and photosystems 1. In cyclic photophosphorylation, it only involves photosystem 1. And that is because, I'm just going to show this over here, I'm just going to rub out the bottom part. All of this text over here. And that is because in certain conditions it might not be appropriate to produce NADPH. This plant might be in a bit of stress. So what happens is that the light energy, it keeps exciting electrons in photosystem 1, and that releases energy to pump hydrogen to activate those H plus pumps and actively transport H plus into the thylakoid space. But those electrons aren't going to be taken up by NADP+. That's because those electrons are going to be recycled over and over again, and by pumping those H plus pumps, we're making small amounts of ATP. And, you know, sometimes plants can go under a lot of stress and it might not be feasible for them to produce any NADPH, so that's effectively what cyclic photophosphorylation is. Alright, I know that's a lot of information to take in. But I'm telling you, as soon as you get the really hard mechanic stuff done, restoration and the rest of the topic becomes a lot easier. So I hope that was helpful. And yeah, I'll see you guys in the next video.